Hi parents, welcome back to biology. Just as a little reminder, you'll be seeing my face quite a bit this year as I hope to give you an up-close and personal look at what your scholar is learning in the classroom. In each of these snack-sized videos, I'll very briefly explain some of the concepts we go over in class, as well as how you can reinforce them at home. I'll introduce vocabulary words they'll be expected to know with the hope that you'll throw them at them at opportune times with you. So like dinner time, weekends, some downtime in the car, give them a word, see if they know it. Each video will also contain a question or two that they might expect to see on a future quiz or similar questions they'll be expected to answer on that end of course exam. I'll also mention any future labs or projects on the horizon for that topic and materials they might need, and each video will have a lucky charm in it. Some extra credit word or phrase, maybe number or topic, will appear at some random time in the video. If students know this word or phrase and apply it on a future quiz, they can get extra credit for it. So definitely stay tuned, parents. As I mentioned in my introductory video, I am excited to partner with you in your kid's success this year. Let's get started. This year, we're starting the year off talking about the nature of science, or how, as scientists, we actually practice designing the controlled experiments that allow us to make discoveries, answer questions, and solve important issues. The scientific method is a loose set of steps that allow us to do just that. In it, scientists ask a question, conduct systematic research related to that question, they form hypotheses about what the answer might be to those questions, and design an experiment that, to the best of our ability, keeps every variable constant and only changes the thing that we think is causing the effect. For example, let's say we wanted to know whether or not scented candles help students perform better on their work, or which brand of detergent is better at removing coffee stains, or which chemical is most effective at curbing the growth of cancer cells. In each case, I'd have multiple groups per experiment. To answer this question, I might have one group of students exposed to the scent of a peppermint candle, one exposed to the scent of vanilla, and one group exposed to no scent at all. To answer this question, I might wash a load of coffee stained clothes in Tide, another load washed with Gain, and then wash a final load with no detergent at all. And for my cancer experiment, I might have a group of cancerous cells exposed to chemical A, a group of cancerous cells exposed to chemical B, one exposed to chemical C, and then one group not exposed to any treatment chemical at all. Now, with each group or load, I would try to expose each of those groups to the exact same conditions. Students in this experiment would be in the same grade, same classroom, doing the same assignments at relatively the same time of day. In this experiment, the loads would be the same size, have the same material clothes used in the same machine, I would have the same amount of detergent, and in this final experiment, I would make sure I have the same type of cells, same temperature, same environment, same amount of chemical, etc. I would make sure that just one variable is different in each experiment, and that variable is known as the independent variable. So in this experiment, the only variable that would be different is the presence of that scented candle. In this experiment, the variable that's different is the type or brand of detergent. And in this experiment, the only variable that is changed or manipulated by the experimenter is the chemical that the cancerous cells get exposed to. Controlling as many variables as possible is the only way that scientists can be confident in the conclusions that they form about the effect that they measure. By the way, the effect or the outcome that we measure is known as the dependent variable. This is always a variable or the thing that we measure at the end of the experiment. So for example, in the candle experiment, it might be the student's scores on that particular assignment or quiz. In the detergent experiment, it might be the number or concentration of stains left on each of the clothing items. And in the cancer experiment, we can measure the average growth of each set of cancer cells after 24 hours of being exposed to that particular chemical. Based on the data that we collect in our experiments, we can form conclusions about how the world works and communicate those conclusions to the rest of the world, especially other scientists. In a process known as peer review, these scientists, including the ones in this classroom, pick apart those experiments and check their conclusions for validity, accuracy, and in some cases, repeat the experiment to ensure that the data is reliable. So let's look at an example of a sample question. A scientist investigates the effect of carbon dioxide on plant height. The scientist grows plants in several closed containers. All of the plants receive the same amounts of water and light, but the air in each container has a different amount of carbon dioxide. What is the independent variable in this experiment? Is it the amount of carbon dioxide in each container? Is it the number of plants in each container? Is it the amount of water provided to the plants? Or is it the height of each plant after a period of time? You can go ahead and pause the video if you need to. Yes, so if you said the amount of carbon dioxide in the container, you're exactly correct. An easy way to identify the independent variable in an experiment is to figure out what is different between each group or condition. In this case, the key word in the question was different. Different amount of carbon dioxide in each container. Students can expect to begin their very first lab of the year this week, in which case they'll be designing and conducting their own experiment that includes all of the variables mentioned in this video. That also means that your scholar will need their lab notebooks, parents, which, if you remember from the welcome video, is just a simple folder. It doesn't have to be 5-star or anything, but a simple folder that has absolutely 100% contains prompts. Well, that's it for our first BioSnack, parents. Thank you again for tuning in and taking an active role in your kids' education. I'll see you next snack time. And don't forget to hit that subscribe button so you don't miss it.